today we're going to have a discussion about antibody and antigen interaction. This is a critical and essential component of acquired immunity, and it's important to understand the relevance and the importance of how this interaction occurs. So the antibody-antigen interaction is what is responsible for a major component of immune reaction to disease. And you can see from this picture here that we have the antibody, which characteristically is shaped as a Y, and it's attached to a pathogen, so to speak, in a very particular way, and the ends of the antibody is very specifically attached to that particular shape on that pathogen. So where do antibodies and antigens belong? They are a component of adaptive immunity, and you can see here that the B cell is the cell that produces antibodies. So the question is, what is an antibody? Antibodies are also known as immunoglobulins, and sometimes you'll see that word used quite a bit in the literature and in other discussions, and it basically means that an immunoglobulin is an antibody. So an antibody is always a protein, and it's a protein molecule that's made in response to an antigen. We'll learn in a minute what antigens are. And when we get vaccinated, what we're doing is we're priming or we're promoting the immune system to produce antibodies against the pathogen that we are vaccinating against. So each antibody that's produced is specific to an antigen. So sometimes you can actually evaluate whether or not somebody's been exposed to an antigen or to a disease by measuring whether or not they have antibodies to the disease. And that's the basis of the HIV test, for instance. So the binding of the antibody, the binding site, is variable and it's configurable so that when an antigen comes along that the antibody has never seen before, the antibody can configure itself to the shape of the antigen. And it's just like a lock and key kind of interaction. So in continuing our discussion of what an antibody is, when an antibody binds to an antigen, another way of saying it is it connects to an antigen, the antigen is then flagged for destruction. And the reason for that is the antibody actually is like a flag or a sign saying to other immune cells that are designed for destruction that this is the place, this is the pathogen to destroy. So the antibodies are produced by, as I said before, the B cells. They're also known as plasma cells. And the antibody production is part of what's called humoral immunity. So every time you see the word humoral, that means antibodies. Okay, and so the production of antibodies is the main function of the humoral immune system. We're going to see that there's another part of adaptive immunity that we'll talk about when we talk about the T cells. So the antibody structure, it's very important. The ends here, these variable regions, that is where the binding occurs to the antigens. And it's variable because these parts of the antibody can change shape based on the configuration of the antigen. So the paratope is what's called the antigen binding site, and that binding site actually binds to what's called an epitope. We'll talk about that in a minute. And the epitope is the characteristic shape of the antigen. So the antibody structure is composed of these light chains, which are in yellow, and then the heavy chains, which are in blue. And they both come together to configure themselves into being able to bind to the antigens. So the other parts of the antibody, we've got this hinge region, And that hinge enables movement of the arms of the antibody so that, for instance, if a pathogen or an antigen is shaped in a very challenging way, the other arm of the antibody can move aside, enabling more room, for instance, for the antibody to bind to the antigen. So these arms then are able to accommodate the complexity and the antigenic molecular structures in order for binding to occur. And if we didn't have this flexibility, we could basically just have one antibody bind to one antigen. Now, the other part is this fragment crystallizable, and that part is fixed, and that part can actually anchor itself into cells, so it's almost like an anchor, so to speak, and it can actually stick right into the cell surface and enable a fixed site for the antibody to actually target the cell in terms of other parts of the immune system. And that part is fixed, as I said, it's not variable, so it cannot bind to antigens. So then the next question becomes, what is the function of antibodies? Why do we have antibodies, for instance? And as I spoke about before, the antibodies provide a very important function. They alert and they recognize foreign proteins and molecules so that other immune cells can come along and eradicate these antigens. These antibodies also activate complement, and in our complement discussion we actually describe that process. So when you review the complement lecture, you'll see that one of the things complement does is it causes cell lysis, which is basically just destruction of the cell, and also it facilitates release of cytokines, which are signaling proteins, and those signaling proteins also serve a function of attracting immune cells to the site, and that word is actually called chemotaxis, where you actually attract things to the place of origin based on chemicals, or in this case, protein molecules. So what kinds of cells are attracted are macrophages, which are a kind of innate immune cell, and T cells, which are also part of the adaptive acquired immune system. So further functions of the antibodies is they bind to immune cells also, and they activate their specific functions. So they can activate platelets in terms of clotting. They can activate macrophages, which are basically a killing or an engulfing cell, and they can certainly do that by opsonizing, which is tagging the antigen, and phagocytizing, which they can actually engulf and kill the cell, but they can also take parts of the cell and put those parts actually onto the cell surface. These antibodies also activate mast cells, which function in terms of releasing histamine, and they cause vasodilation of the blood vessels. And also, very interestingly, they interact with these receptors that are actually on the placenta of a woman who's pregnant, and it facilitates the transfer of antibodies across the placenta to the developing fetus. So we know, for instance, that maternal antibodies are transferred across the placenta, and they actually stay in the fetus, and after birth they serve a role in terms of protecting the newborn from infections and so forth. So there are different types of antibodies, and that's what this couple of slides are going to show. And these antibodies have different functions. So the first type of antibody is called IgM, and that stands for immunoglobulin M. And the way you can remember the function of that M is that it stands for memory. And we'll talk about this a little more when we talk about the general functions of the acquired immune system. So IgM is a memory immunoglobulin, and you can see that the IgM is a very complex molecule. It has many arms, many places for those shapes to be configured to antigens. And whenever I explain this concept to students, I speak about if you've gotten locked out of your apartment or out of your house, and you call the locksmith, and they come to your door, and they have a very big ring with all these different kinds of keys, and then they figure out what kind of door you have and what kind of key type you might have, and then they go off and make the shape of that key to fit your lock. Well, that's exactly what an IgM molecule reminds me of. It's almost like a keychain with all these multiple keys. And each of these places can serve a role of being configured to fit an antigen. So sometimes when you have a super infection, you actually have every single place of that IgM molecule occupied with the shape of that particular antigen. So these IgM molecules, so to speak, are on the B cell surface, and the M part stands, as I said, for memory. So after an infection, what happens is these IgMs stay around after the infection, and they allow the immune system to remember the antigenic shape, because their variable region here has been configured to represent that shape. Now, IgM is not perfect, so sometimes, for instance, we'll forget an infection, so we'll actually lose all our IgMs, and then we're actually prone to get reinfected, excuse me. So also IgM in a new infection is the first antibody to be produced, and it causes an immediate reaction. It causes also a short-term immune reaction, but then, as I said, it has the memory function, and it can also stay around at the long term. Now, the other molecule that stays around for the long term is the IgM, the immunoglobulin, or not IgM, excuse me, the immunoglobulin G. And that antibody is the most concentrated in serum, and it works in concert with IgM. So it's easily diffusible, and excuse me, it's the second antibody produced in an infection, and it works with IgM to produce this lifelong immunity. So if you do, for instance, have this lifelong immunity, so another way to think of it is actually to think about, why do we get vaccinations? Well, we get vaccinations to essentially teach IgM and IgG about this pathogen, so that if we truly get infected, we'll already remember the infection and we'll be able to launch an immune reaction much, much quicker. So really, that's really the purpose of getting these vaccinations. So each IgG antibody, for instance, is specialized in terms of shape to a specific bacterium and its components. So let's go on and talk about the other immunoglobulins. So we have immunoglobulin D, and we really don't know that much about IgD. It's a maturation marker. It's also on the B cell surface. It facilitates the B cell responding to a T cell signaling. But again, it's not fully characterized. The function is not fully characterized. We have immunoglobulin A, which provides protection at mucosal surfaces. It's also found in the first secretion of mother's milk at birth of an infant. And then finally, IgG, this is the antibody that's often involved in allergy and allergic reaction and asthma. It's also affiliated with the mast cells and with basophils and with eosinophils, and we're going to learn about these cells in the innate immune system. And it also serves a role in terms of having a response to parasites. And again, it also has this autoimmune process, which we'll talk about in another lecture. So to continue our discussion, we want to ask ourselves, well, we've learned now what an antibody is. Then let's talk about what an antigen is. Now, the thing is about an antigen doesn't necessarily need to be a pathogen. For instance, it doesn't need to be a protein. It can be any kind of molecule, actually. And usually these are small molecules in size. The definition of an antigen is to actually provoke an immune response. So if the antigen does not provoke a response, it's not really considered an antigen. However, some people, if they're immune suppressed and an antigen, for instance, induces a response in some individuals but not others, that antigen would still be called an antigen. So the antigens cause or induce production of antibodies that are specific, remember that word, specific to the particular shape and structure of the antigens. So there are several types of antigens dependent on what their origin is or what their source of origin is. So we have exogenous antigens, which are the most usual kind of antigens, and exogenous means that the antigen comes from the outside. So they can have certainly an environmental source. They can be ingested, inhaled, injected, or absorbed through our various, remember, mechanical barriers. They can be viruses, bacteria, some kind of chemical, or a fungi. We can also have what's called endogenous antigens. Now, endogenous by definition means within. So it's antigens that are found inside of the human body. Now, these endogenous antigens can be actually inside of the cell, and they can be components of the host itself. They can be bacterial or viral pathogens that release, for instance, toxins inside the body. And when these things are released from the cells, these antigens are presented by these MHC1. Now we'll learn about what that is, major histocompatibility 1 molecules, and then they stimulate an immune response. Now the autogenous, what that antigen has to do with autoimmunity, which we will have a separate lecture on autoimmunity. And that's a very important disease process where the human host recognizes itself as being foreign, and it's considered dysfunctional, and some component of the human host, some cell or protein or molecule that originally was seen as part of the body is seen as foreign, and that antigen then is treated or recognized, and we have antibody production, and we launch a complete immune reaction against something that may not truly be foreign but be part of the body, but that whole process is dysfunctional. So in terms of now that we've learned about antibodies and antigens, let's talk about how they react. So we have different types of interactions. These types depend on the strength, on the number, on the combined strength. So sometimes individual interactions are weak, but together they're very strong. So the concept of affinity means the strength of the interaction between one single antigen and an antibody. So we talked about the antigen having a very special shape, and that shape is called an epitope. and the antibody having a complementary shape called paratope. Avidity means that the combined strength of a single antibody, where all the antibody positions, so to speak, are occupied. So we saw that IgM has a total of 10 antigenic sites, whereas IgG has 2. So I talked to students about another example of IgM is if we think of IgM as an apartment building, and there's 10 units, so we can have different pathogens living in each of the units, or we can have the same pathogen in all of the units. And if we have that situation, then that's a very, very strong signal. My God, the message to the immune system is remember this pathogen. Don't ever forget it. And that's what happens when IgM, for instance, has each individual site occupied with the same pathogen. So to continue our discussion, there are other types of interactions as well. The concept of specificity that we discussed earlier has to do with one type of antibody being configured for one antigen. And that can be thought of as a very specific interaction. And that's different from this other concept called cross-reactivity, where we have two similar organisms producing antibodies and then they cross-react. So an example of that are antibodies against the West Nile virus and antibodies against yellow fever. These two viruses are both carried by mosquitoes, and the antibodies of these diseases, so to speak, actually cross-react. So for instance, if you're exposed and come down with West Nile virus, and then later you're exposed to yellow fever, you could have a cross-reactivity situation where the antibodies of one may protect you from the other. So now we come to the question of antigenicity. So how do the antibodies recognize, or how well do the antibodies recognize the antigens? So the antigenicity is the ability of an antigen to elicit an immune response. So depending on these various concepts will tell you whether or not you're going to get an immune response from a particular antigen. So one concept is foreignness. So in order to get a reaction, so to speak, the phylogenetic distance or genomic similarity. So the distance, another way of saying the similarity, needs to be far apart in order for the immune system to recognize that antigen. So smaller genetic distances means that there's a closer relationship potentially between the antigens, and then you're not going to get an immune reaction. So that's the reason why often when individuals need a transplantation, that they go to their immediate family. So for instance, to get a bone marrow transplant, you need to make sure that the donor is very genetically similar to the recipient. And if the donor is very similar to the recipient, then you're less likely to have rejection. Why does rejection occur? Rejection occurs because the immune system recognizes the transplanted tissue as being foreign and then launches an attack against this foreign tissue. So in the case of transplantation, you want to have the transplanted tissue be very closely related genetically to the donor. So the donor and recipient, to have it work well, needs to have a close genetic relationship. Another determinant of antigenicity is size. So the bigger the antigen, the more easy it is to recognize it. So these large structures are more easily detected by immune surveillance. Now, we have very, very tiny viruses, and we'll see later on in the virology discussions that, you know, we want to make sure that these viruses do not escape detection. So that's a big issue that we'll review when we get to the virus lectures. So the next question we need to address is what determines antigenicity? And really, antigenicity is the capacity of the immune system to recognize the antigen. And so, you know, commonly, characteristically, the more different an antigen is from the immune system, the more recognizable it will be. And so some of the criteria include conformation. What is the shape of the molecule? That's really important. And remember, the protein molecules are the ones that characteristically can become very complex in shape. You know, primary, secondary, tertiary, quaternary, these are the ways in which protein can reconfigure itself from being a simple linear amino acid chain all the way through to being a quaternary structure. Composition, protein amino acid sequence. What is the amino acid sequence? Is the composition. Is it a double-stranded DNA? Is it a double-stranded RNA? Is it a single-stranded RNA? What are the composition? What are the nucleotide base composition of both molecules? Obviously, that's going to make something different from something else. Bacterial components. Are we talking about a cell wall? Is it gram-positive? Is it gram-negative? Flagella. Does the bacteria have a flagella? Mannose, what sorts of lipopolysaccharides are sticking outside of the cell wall of the bacteria, for instance? These are all things that trigger immune system response. So, what are some specific responses that happen as a result of these antigens? Microbial cell wall components. So microbial cell wall components. What molecules compose the cell wall? What does that trigger in relationship to the immune system? Phagocytosis, complement activation. Remember, complement activation had those pattern recognition capacity, or the capacity to recognize those patterns, and certainly how the microbial wall, what it's composed of. Remember, the lectin complement pathway is based on lectin molecules basically sticking out of the bacteria. Lipoproteins. Remember, lipo is a lipid attached to a protein. The gram-positive bacteria. Remember, gram-positive bacteria have a cell wall on the outside. These lipoproteins recruit macrophages, and they cause release of cytokines. What about double-stranded RNA? Double-stranded RNA. What does double-stranded RNA do? It triggers antiviral cytokine release in relationship to interferon. Remember, we have interferon, we can have interleukins, we can have other kinds of cytokines. Double-stranded RNA, obviously that needs to be a virus, and it's an RNA virus, right? So the RNA viruses trigger very specific antiviral cytokine release. What about lipopolysaccharides? Now, remember, the lipopolysaccharides are found in gram-negative bacteria, and gram-negative bacteria, remember, have the cell wall on the interior, and we have that cytoplasmic covering that contains a lot of things like enzymes and lipopolysaccharides and various things that can actually combat these different mechanisms that the immune system utilizes. So these lipopolysaccharides also recruit macrophages and elicit cytokine release. Now, what about flagellin? Well, flagellin is what these flagella are made of, okay? So we can have a bacteria with that flagella, and that flagella is actually the antigen. So the flagella, the shape of the flagella and the fact that it's composed of flagellin, that's what elicits the macrophages to come on the scene, and it elicits release of cytokines. What about now? We can have, of course, RNA is usually single-stranded. Uracil-rich, single-stranded viral RNA. What does that elicit? Antiviral cytokine release in the form of interferons. So you can see that there are quite a lot of different types of antigens, and they're composed of different things, and they elicit different kinds of responses in the immune system. And we have been talking not about acquired immunity, but innate immunity. So this is even before we get to the T cells and the B cells. So the first question we have is, we learned in the tumor immunogenicity lecture that tumors can escape detection by the immune system. What things can we do to enable the tumor to be recognized? So number one, tumors are part of the human body, right? So in early stages of cancer, right, the cancer cells have not become that different from normal. So in the beginning stages of tumor development, it's no wonder that the tumor would escape detection. But sometimes the tumor actually has mechanisms and things that are secreted that cause immune suppression. And sometimes in the case of this long-term chronic inflammation, for instance, in that situation, kind of naturally, the immune system is unable to recognize these tumors, and especially these growing tumors. So one thing that's going on, one line of research in tumor biology is actually injecting. So you can actually inject a tumor. So here's our tumor. You can actually inject actual, like, benign viruses that are recognized by the immune system. So, say for instance, we inject some benign virus into these tumors, and then all of a sudden the immune system now recognizes the tumor because it recognizes the virus that was injected into the tumor. Now, there are other kinds of research going on that can actually cause tumors to become sensitized to chemotherapy by, injecting other things and enzymes into tumors and so forth. But really, there's a lot of work going on now in tumor immunology where we're developing ways to kind of jumpstart the immune system or get the immune system in some kind of way to actually recognize the fact that a tumor is growing. So these are some of the things that we can do in relationship to getting the tumor to be recognized. Excuse me. So the second example, an antigen can be almost anything, right? It doesn't have to be a protein. It can be, a lipopolysaccharide. What's a lipopolysaccharide? It's a fat molecule connected to a carbohydrate. Or the antigen can be a collection of molecules, or it can be a whole structure like a flagella, for instance. Why? So the fact is that antigens can be anything. However, antibodies must be made of protein. So you ask yourself, why is that? Well, we kind of brushed on it a little bit, that actually, remember the proteins. We have a primary protein. We have secondary, tertiary, and quaternary. And the elegance of these proteins is they're very malleable. They can be shaped into very complex structures. And they have, and what's the other, obvious major issues about protein, major quality or characteristic about protein It is made, right, from DNA, transcription, and translation. Remember, translation by RNA. Okay, so if we have a brand new antigen that the immune system has never seen before, right, how can the immune system figure out the shape? Well, one thing it can do is it can create antibodies that help the immune system recognize the antigens, and these antibodies can also take out the antigen. So what we learned is that these antibodies, right, can also configure themselves to shape, to be a complementary shape to the antigens. And it does this through this whole reconfiguration of the DNA in such a way that it can construct new antibodies that the immune system has never seen before. So we can't have antibodies made from, let's say, lipopolysaccharides, because why? Because we don't have DNA for lipids and we don't have DNA for carbohydrates. We only have DNA for protein, and that we can reconstruct almost using protein into almost any kind of shape that the antigen might present itself with, or might be presented to the human body. So this is really key. Antibodies are always protein. So if you have a multiple choice, what are antibodies? Pick protein. All right, so let's go to example number 3. How foreign does an antigen need to be in order to be recognized? Okay, so what does that mean, how foreign? Well, it has to be foreign enough, right? But the key word is enough. Now, it's relative, isn't it? Because what we know, for instance, say if indeed you want to get, you need a transplanted organ, for instance, say you need, you've been suffering from diabetes, unfortunately, and the kidneys have gradually deteriorated over time because of that. And at some point, you need a kidney transplant. So, what do they do? Who do they look for? And why? Okay, so here's the question. What is the big issue about getting a transplanted organ? The big issue is you don't want rejection. You don't want the immune system to recognize that organ as being foreign. So often what happens when you require a transplanted organ is that the first thing that happens is they look at the relatives of the individual who needs the transplant. Why? Because the relatives are very genetically similar to the recipient, and the more similar you are in terms of that transplanted organ, the less likely you're going to have tumor rejection. So basically, with this transplantation, what you're trying to do is you're trying to find an organ that is not foreign from the human body. But foreignness can also indicate, is the tissue, or is the pathogen, or is the antigen non-human? Is it from a different species? Now, the other thing we know is that it's easier, let me make sure that's spelled, easier for the immune system easier, let's put that in quotes, easier for the immune system to recognize a eukaryote. What is a eukaryote? Well, a eukaryote is a cell with a nucleus, right? And isn't the human, oh gosh, here I am. I'm even missing my own question. Actually, it's easier for the immune system to recognize a prokaryote. Okay. And why? Because a prokaryote is a cell that does not have a nucleus. What is the number one prokaryote pathogen? Prokaryotic pathogen, it's bacteria. Okay, so it's easier for the immune system to recognize a cell that is not a eukaryotic cell. It is more difficult, let's put that in quotes, for the immune system to recognize, it is more difficult for the immune system to recognize a eukaryote. What are some eukaryotes that the immune system has a difficulty recognizing? Fungi, helminths, and the third kind is the protozoa. Now, there are lectures for each one of those organisms, so I highly recommend you refer to those lectures to get a better handle on how the immune system recognizes those organisms. So, in relationship to answer, how foreign does the antigen need to be recognized? It needs to be foreign enough. And this whole lecture, at least is in relationship to antibodies and antigens, is really devoted to that particular question. So that ends our lecture for the day. Thank you so much for visiting educator.com.